Hello, the purpose of this video is to help you guys to get prepared for the cellular respiration lab that you're going to be making inquiry this year. And in order to do that, I need to tell you the basic structure of what would be done with a respirometer so that you understand how a respirometer works. And then from there, you can figure out how you can manipulate certain environmental factors that might affect the rate of cellular respiration. Ultimately, that is what you need to be testing. You need to be testing the impact of some factor on respiration rate. Okay, so you will be calculating rate by the time we're finished with this. This lab sheet is on Google Classroom so that you can take a look at it, but once again, this is the lab sheet that was utilized last year and many other previous years. Doesn't have an inquiry component to it. We're adding that this year. So reading through this will give you maybe some ideas for how this should be done, but you ultimately have some choices to make. Okay. So there's some background information, some objectives. You can take a look at those things. I'm going to be mentioning them kind of as I go through it, like the gas law is that PV equals nRT that you learned about in chemistry class. There is an important thing to understand about this process that does relate to that equation. So I'll be coming back to that when it applies. Okay. There's talking about PV equals nRT. Okay, so the general setup for a respirometer, let me get to the draw, is that you're going to have, a vial, and in the bottom of the vial, you're going to put a cotton ball. Okay, so that's cotton. And in the cotton ball, you're going to put a chemical that is called potassium hydroxide. Now, potassium hydroxide is caustic. It's something that when you're working with it, you need to have goggles on. When you're getting it set up, different people should have different roles, and the person that is working on getting this component of the vial set up needs to be wearing goggles. The point of the potassium hydroxide is that it will take the CO2 that's produced in cellular respiration and produce a solid out of it. So it takes it out of a gaseous state and puts it in a solid, which is much, much, much more compact, which then allows us to basically ignore it as far as the volume is concerned. So then what you can end up doing then is really directly see the amount of oxygen that's consumed by whatever critters that we put in there. The volume of the bubble that's going to be inside here will get smaller as oxygen ends up being consumed. If we didn't absorb the carbon dioxide, the amount of oxygen consumed would just be replaced by carbon dioxide produced. So it's very important to have this carbon dioxide absorber in your cotton ball at the bottom. The thing, though, about these potassium hydroxide, like I said, it's pretty caustic, so we don't want our critters, whether you're working with peas or whether you're working with the mealworms or something else, you don't want whatever this critter is to be sitting right on top of the potassium hydroxide. It's not nice stuff. So in between, you're going to put a very thin layer of rayon. Rayon, it kind of looks like a cotton ball, but it's non-absorbent, so the liquid won't pass up through that. So you're going to put a little, that's like a divider. It's a little separator, but like I said, it's like a cotton ball, so certainly air can pass through that. You need that to be able to happen. All right, so then you're going to put whatever critters you're going to put in here. So let's say that you're going to put peas in here. I'll do that just kind of as the standard setup, and you guys can consider what you're going to put in. So if you're going to put peas in, the setup was to put 15 germinating peas into the respirometer. And then you're going to cap it off with a stopper that has a pipette on it, and this pipette is graduated. So what you're going to end up then doing is eventually sinking this in a water bath, so it's completely underwater, and so you're going to have this bubble of water that's inside of the of the respirometer, and that bubble of water is going to get smaller and smaller and smaller as time goes on because oxygen is being consumed. So you're going to make measurements on this pipette as the water moves down toward the respirometer. It is a very, very delicate experiment, so you need to make sure that you're reading that pipette carefully to the correct number of significant digits. So take a close look at it. The graduations on it are in tenths of a milliliter. So the lines in between then would be hundredths. So then you would be estimating to the thousandth of a milliliter. All right, so that's kind of the general setup. But in each container, you also are going to need to have a control. And the reason we need to have a control is because anytime there's any changes in atmospheric pressure or temperature changes because of PV equals nRT, that can affect the volume of the enclosed bubble. The volume of that can be affected by pressure changes and temperature changes. So keeping temperature in your water bath constant is a very important thing to do. And this control vial will be important for you to be able to subtract off any changes that happen in your control vial are not due to respiration, so you can subtract those differences off from your experimental vial. So the control vial will have everything the same, so it's going to have the cotton in the bottom, and it's going to have the potassium hydroxide in it, and it's going to have a rayon divider in it. But instead of having your peas or your whatever you decide to put in there, instead of that, we're going to put in something that occupies the same volume but does not respire. And so you need something in there that's taking up space, but it's like glass or plastic. So I have these blue circular, they're just beads. That's the standard, what we've been using. They kind of are pea-shaped and sort of pea-sized. But you need to make sure that this volume occupied by those glass beads or plastic beads is equal to the volume occupied by the peas themselves. So the experiment calls for using a known volume. So the first thing you'd have to do while you're getting this set up is you need to find out what is the volume that's occupied here. And in order to do that, I recommend using displacement. So while you're getting this set up, you'd get a graduated cylinder. You would put in, 50 milliliters of water. Then you plop in your peas. That volume goes up, and then the difference between this is the volume of the peas. And then you would do the same thing for your beads. Start with the 50 milliliters and add beads until that volume that goes up is equal. So you've got this equal amount of space occupied there. This is a challenge if you are using the mealworms. It's a challenge because I'm not going to let you use displacement on a living thing in order to figure out its volume. And so you're going to need to use your skills of math in order to figure out what is the volume of a mealworm or two. I don't know how many mealworms you're going to decide to put inside of each one of your containers. And then you need to have still the same amount of volume occupied in your control respirometer using those glass beads or using something else that is just occupying volume but certainly not respiring. Okay, now when you get your respirometers all set up, you're going to need to get them acclimated into the water bath. The lab protocol calls you for putting them in the water bath with their tips just sticking out of the edge for a period of time, about 5 minutes to do that. And then what you should do in order to make sure that it's a little bit easier to follow the water as it's getting sucked down through the pipettes, to add a little drop of food coloring right at the tip of the respirometer give that just a little bit, like a minute, 30 seconds even, and then you're going to sink the whole thing. So then the whole thing will end up being sunk underwater. Leave it sit there for a minute or so to equilibrate. And then after that period of time, you do not want to touch these respirometers at all. It's very sensitive. Any waves in your water bath are going to definitely impact because pressure certainly is an important thing. So be very, very, very careful. Now, when you guys are doing this, I want you to have a control in each water bath. You need a control for every water bath that you've got, because the temperature changes in one water bath might not be the exact same as the temperature changes in another water bath, so they each need their own control if you have more than one water bath. And then I want you to have as many experimental respirometers as you can. I think the limit that we're going to end up running into for this lab is that we're going to run into it being difficult to get it set up fast enough. So you need to make sure that you really, really, really know what you're doing on Friday when you come in. People with first hour, you might want to see if you can come in a couple of minutes early to start to get this set up, because you do want the respirometers sunk in that water bath for as long as possible. I would say we want them in there for at least 15, maybe 20 minutes in order to really see any kind of a difference. So those are my recommendations. I do want you to have a comparison between two different experimental treatments at least. You could do 3, but then you need to make sure that you can produce averages of them. So if you're going to do 3, then you may end up having to you have to figure out how they're going to be put into the water baths.